You're welcome to Learn at Home live session. It's going to be an exciting moment together with you. My name is Samson Bolarinwa. I'll be taking you through biology today. Call your friends. Get your learning materials. Sit down as we learn together. Today, we'll be looking at a very interesting topic. Genetics. This topic, genetics, will help us to provide answers to some of the questions that we must have been asking. Questions such as, why are my classmates, why are some of them tall and some short? Questions like, why do we have some dark-skinned people and some light-skinned people? Questions like, why do we have some plants that are very tall and some are short, though they belong to the same species? The concept of genetics will help us to understand this. Why you look like your parents, why some people don't look like their parents. These are the questions the concept of genetics will help us to provide answers to. So sit back, relax, as we learn together. First of all, we'll be looking at the definition of genetics. We'll be learning what it means, the meaning of genetics. And after that, we'll be considering different terms associated with genetics. Genetics as a concept consists of different terminologies. These terminologies are very important. You must understand them, and with your understanding of these terminologies, you'll be able to answer questions under the topic. And finally, we'll be looking at the Mendelian laws. Mendelian laws. Don't forget, the last time we had our biology class, we considered evolution, and there we also learned theories of evolution. So today we'll be looking at Mendelian laws of inheritance. So sit back, get your learning materials as we learn together. Now let's start with the definition of genetics. What exactly is genetics? Genetics as a concept, what is the meaning? Genetics is a branch of biology which studies two major things, heredity and variation. Don't forget, in your SS1, you must have been taught the branches of biology. They are numerous. Remember zoology, remember botany, remember physiology, remember entomology, remember different ones, physiology, that you have been taught. So genetics is just a branch, just like those ones. But genetics as a branch of biology is addressing two major things. It's the study of heredity and variation. Now we'll be looking at the meaning of these two concepts, heredity and variation. When we talk about heredity, what do we mean? Heredity is or we call it inheritance, is the transfer of characters, traits, from parents to their offspring. What is this term talking about? Heredity is talking about how those parts of your body that look like that of your parents, how they are being transferred to you. Take, for instance, if you have the same nose shape as that of your parents, how is it transferred from your parent to you? That's what heredity is trying to address. Then variation is trying to talk about the differences that we have among the parents and offsprings of the parent. You know, when you have children from the same parent, though we may have similarities that they have, however, we have also some differences. So the concept of genetics is actually addressing two things. Why you look like your parents and why you don't look like them. Whichever way, whichever category you fall, that is what genetics is trying to address. So again, genetics as a concept is a branch of biology which studies heredity and variation. Heredity and variation. And scientists who study genetics, they are known as geneticists. You know, geneticists, they are the group of people that studied this concept called genetics. Probably after this lesson, some of you will develop interest that you want to become geneticists. You want to study more on heredity and variation. So if you are saying you are studying anything on genetics, you are studying two major things, heredity and variation. Don't forget, heredity is the transfer of characteristics from parent to their offspring, while variation is the differences we have. Then we move on to the terminologies. What are the terms that are used in genetics? Like I said, this is key. Your understanding of this concept is what will help you to answer questions in genetics. Once you don't understand this concept, you may find it difficult to answer those questions. So I need you to sit down, relax, pay attention as we go through this together. Now let's look at some of those terms. We have genes, we have chromosomes, we have characters, we have gametes, we have allele, we have phenotype, and we have genotype. Dominant character, recessive character, homozygous, filial generation, Aploid, Dihybrid, Testcross, and Backcross. These are some of the terminologies we'll be considering. Once you are able to understand it, then you'll be good to answer any question that will come your way in genetics. So let's look at the meaning of each, one after the other. To start with, we have the first one, genes. Earlier I told you about heredity. That has to do with what you have inherited from your parents. Now there is a part of your body, there is a part in your cell that helps to transfer that trait, that character from your parents to you. That is called gene. So gene is that part in the cell that helps to transfer traits, characters, features from your parents to you. Maybe your height, for example, is a trait that you have inherited from your parents. There is a particular gene responsible for that feature. So that gene is what was transferred to you that makes you to have that particular feature. So gene is a basic unit of inheritance that is responsible for transmission of characters from parent to their offspring. So that's what gene is. The next concept is chromosome, is another term. Chromosome is a rod or thread-like bodies that is found in the nucleus of a cell. I hope you can remember the diagram of your plant cell and animal cell. When we talk about plant cell or animal cell, one of the organelles, I mean the parts in the cell that you will see when you draw it is nucleus. This nucleus is common to both plant and animal cell. Now, why do we need to talk about chromosome? Since we say it is just a rod or thread-like. This chromosome is very important because it is the one that is housing the gene. Don't forget, gene is that particular part of the cell that helps you to transfer features. So it's a basic unit of inheritance. Now let's take, for instance, if this is our cell. This is our cell. If this is our cell, in each cell we have what is called nucleus. We have nucleus. So let's take this as our nucleus. In the nucleus, we said we have a thread-like or rod-like body called chromosome. Okay, let's use this so that we can see it clearly. Now, chromosome. Then in the chromosome, that's where we have what we call gene. So you can see that if you want to get to the gene, you first of all get to the chromosome, you get to the nucleus. So if you are coming from outside, you get to the cell, from the cell to the nucleus, to the chromosome, before you can reach the gene. So it's a structure like this. So the chromosome is the structure that helps us to house the gene. Then we talk about characters. Look at this picture. It's showing us the relationship between chromosome, nucleus, and the gene. So we talk about characters, first of all, so that we can understand it. When we talk about characters, we have been talking about the inheritance of characters. When we talk about characters in genetics, we are not actually talking about behavior like our day-to-day English. In this concept, when you hear the word character or traits or features, we are talking about that feature that can be passed from parent to their offspring. It could be height. We have some of the characters now. When we talk about characters, these are things we are referring to. It could be height, it could be shape, it could be color, it could be size. So these are things we refer to as characters that are passed from parents to their offspring. You need to know this. A character that is passed from parent to offspring is controlled by gene. Like I said, you must understand it. Now, each character is controlled by gene, but each character that we have is controlled by a pair of genes. Let's say, for instance, we are talking about height now, tallness. When you talk about a character is controlled by a pair of genes, that's true. Now, this can be either dominant. The character can be either dominant or recessive. What do we mean by dominant character? Now, dominant character is actually saying the character that expresses itself over the other character when both of them are present in a particular situation. So, in genetics, you need to take note of this. When you see capital letter or uppercase, we use it to represent dominant traits. We use it to represent dominant traits or characters. And when you see a lowercase, we use it to represent recessive traits. So we don't just use the letters like we use it in English that are at the beginning of the sentence. No. When we talk about, when you see this uppercase in genetics, we use it to represent dominant characters. When you see lowercase, we use that to represent recessive characters. That will take us to another terminology, and that is Aploid. Aploid. When we talk about haploid, it's a cell that contains a single set of chromosomes. A cell that contains a single set of chromosomes. Now, chromosomes are in sets. You know, it's like you are saying package. They are in sets. So when a cell contains just a set, we say that particular cell is haploid. A set is usually 23, and we represent haploid using small letter n, 23 pairs. And the other concept is diploid. Diploid, as the name is, di, meaning it has two sets of chromosomes. 2 sets, and that's 46. So this one has, in the set of chromosomes, it's made up of 23, while diploid has 2 sets, meaning it's going to have 46. So that's what we mean by haploid and diploid. This will help us to understand this concept. When we start applying them, you will know why we have to learn this. Okay? So that's for haploid and diploid. Then we look at another concept, allelomorph. That particular word, or we call it allele. Allele. That particular word is just giving a name to those genes. For example, initially I told you that each gene is controlled by a pair. Each character is controlled by a pair of genes. Now, each of these letters is what we just give a name to refer to as allele. It's talking about the variations of genes. That's what we refer to as allele. Some of you must have done genotype tests at a point where you see, oh, you say I am AA. This one will say I am AS. Now, these are alleles. Each of them. So we have this is an allele, this is an allele. So different variations. For example, for tallness, we can have TT, or we have this. This is for shortness. So this is an allele. Now, when you now talk about different versions, someone may have, or an organism may have something like this, or this. This is talking about different versions now that you can have, or this. And take note, one more thing, when we are writing these letters that we refer to, the capital must come before the small letter, because the capital means dominant. So it must come before the recessive. Now, how do we know if it is dominant or recessive? Let's take, for instance, we have two organisms. One is tall and one is short. This will help us to understand another concept, which we call homozygous. Homozygous. When we say homozygous, what we are talking about is the allele, they are of the same, not different. Meaning, you have capital T, capital T. It's a pure breed. Is that we call it homozygous, or we call it pure breed? Because it's made up of two similar letters. This is also homozygous because they are both lowercase t's. So this is also homozygous. Now, the other one is heterozygous. When we talk about heterozygous, we are talking about two different alleles. Now, it can be this. So this is an heterozygous gene. How do we come across this? Let's quickly look at how we come across it. We come across these things using what is called genetic diagrams. Genetic diagrams. Now, in genetic diagrams, we can do it in two ways. For example, if you are given that we have two plants, two parent plants. Parent plants. One is homozygous tall. Don't forget homo, the same. And the other is heterozygous tall. Different letters. Capital must come before small letter. Now, if you have to use your genetic diagram to do this, you put it this way. First of all, you write parent, T, T. T, T. Then you go ahead to write G. G here is talking about gametes. That is what will be contributed during reproduction from male and female. Like in human now, during reproduction, the male gamete is sperm, while the female is ovum. So that's gamete. When it becomes gamete now, you space this out because you want to do your genetic cross. Don't forget the question given to us, we have two parents. This one is homozygous tall, and this is heterotall. Heterozygous. Different letters. Now we want to see the kind of offspring that they will be having. What you do is you take this one after the other. This sign here is representing fertilization. So we take it one after the other. From the first T, you go to this one. So we have TT. Then you go again to the second T. We have capital small. Then you do the same, but you do it to this direction now. This is TT. Then we have to this TT. So in this one, what we are saying is this. These are the parents. Homozygous tall, heterozygous tall. This is called F1. F1 is telling us about filial generation. What we mean by filial generation is talking about the children, the offspring that they will have. The first set of offspring. It's as if you have father and mother. And the first set of children is the first generation. So we call it first filial generation. So that's the meaning of F1, filial generation. Now, this will help us to understand another concept. In this F1, you can see that these parents, they have given birth to 4 children. Now, out of these 4 children, the question is, how many of them are tall? And how many of these children are short? If you look at the parents, we said this one is tall, but homozygous tall. This was also tall, but heterozygous tall. Now, compare it with this result. What will you say about this firstborn, the first child, tall or short? Tall. The second one, tall or short? Tall. The third one, tall or short? Tall. The fourth one, tall or short? Still tall. But when you look at it, you will see that we have 2 of them that are homozygous tall. They have the same. Meaning, in them, they have gene for tallness, no gene for shortness at all. That's what we call homozygous. They are called pure breed. Pure breed. But when you look at this second one and the last one, they are also tall, but they are heterozygous tall. Hetero. So when we say homo, it means you have the same allele. When we say hetero now, different version. So they are all tall, but we have homozygous tall and heterozygous tall. This will also help us to understand another concept, and that concept is phenotype and genotype. When we talk about phenotype, when you look at these children, we agree that they are all tall because the capital, once you see a capital T beside a small t, you know we have said that the capital represents what? Dominant gene. Dominant gene. So, and once they are together, the dominant gene is what we mask the recessive gene. That's why we call them dominant. It's like you're having 2 people fighting, and one is stronger than the other. So once the one that is stronger is there, the one that is weak will just keep quiet. So that's what this is talking about. So in this one now, there is no gene for shortness. So this is homozygous, but there is a gene for shortness in this particular organism, but it's still tall, but carrying the gene for shortness. That will help you to understand. So if you look at this physically, we say they are all tall. They are all tall. So that's what we call phenotype. So if you ask now, what is the phenotypic ratio of this? We'll say it is 4. But when you are now asked, what is the genotypic ratio? Now, when we talk about genotype, it's not what you see. The fact that you see someone that is tall, you cannot tell if the person possesses a gene that is the shortness gene or not. But you know the person is tall. But there is tendency for a tall parent to give birth to a short child. What is responsible? There is every possibility that that particular parent is having a gene for shortness, but it is being masked by the gene for tallness, which is the dominant gene. So in this case, the genotypic ratio will be 2:2, because we have 2 homozygotic and 2 heterozygotic. So we just say the phenotypic ratio is 2:2. So I hope you understand these terminologies that we are looking at. Now, having explained these terminologies, let's quickly move to other aspects. We have explained filial generation. By now you know what it means. Then another thing is hybrid. When we talk about hybrid, you must have been hearing hybrid. It's just an offspring from a cross between parents that are genetically different. And that's what we did. That was what we did. This is a tall parent. This is also a tall parent. Phenotypically, that is physically, when you see them, you say they are tall. But this one is a pure breed, homozygous. It doesn't have any gene for shortness. But this man is tall, but there is a gene for shortness. So when we say that, now, when we talk about hybrid, it means that we want to cross 2 different organisms. They are of the same species, but they are genetically different. So these ones are genetically different. But when you look at it phenotypically, that's the physically, they are both tall. But the gene now, this one has gene for shortness, while this one does not. So an hybrid is the cross between 2 offsprings of the same species, but different genotypes. Do you understand that? So that's what we mean by hybrid. Now, the next thing is backcross. We have a concept we call backcross. Backcross, as the name implies, is a cross between the offspring of a parent with the recessive, homozygotic recessive gene of that parent. What do I mean? Let's say we have this homozygotic tall father, and the mother is short, homozygotic short. Meaning the father is a pure breed, has no gene for shortness, while the mother is also a pure breed for shortness. She doesn't have any gene for tallness. Now, when they give birth, what are the children they are most likely to have? Don't forget, when you are doing your crossing, you have P, then you have G to show the gamete. Now, this is parent. The gamete, when you get to your gamete, you space it out because you are going to cross. And don't forget to put your fertilization or meiosis there. Now, what are the kind of children they are going to have? Take this. That's the first one. Take it again to this. Okay. Then we do the same thing. Do it towards this area to make your work very neat and not to confuse the examiner. Now, when you look at it, all the children they have, they are tall phenotypically. If you ask me, phenotypically, because you can see when you see dominant gene. However, the kind of tallness is they have heterozygous tallness because there is a gene for shortness in each of them. In each of them. So when you are asked to give a back cross, meaning go back to the parent and look for the one that has homozygous recessive. 2 terms. Homo, meaning it must be the same. When you look at it, homo, they are both homozygous. But the question says recessive. This is dominant. Don't forget, capital letter, dominant, and small letter, recessive. Now, the recessive one, that is homozygous recessive, backcross means take it and pick one of these children, cross it together, and see what you are going to have. So if we are to do that, remember the genotype of the children, capital T, small t. So we take that to see the kind of children they will have. So let's look at it now. So this is the child now, and this is the homozygous recessive parent. So we cross them too. That's what backcross means. When you hear backcross in genetics, don't forget that's what it means. Now we have this. Let's see what we are going to have in this case. Okay. Now, look at the children. Don't forget the first one was the F1 generation. We picked this one from F1 generation. So this one will give us F2. That's second filial generation. When you look at it, Backcross now is giving us 4 children, and out of it, we have 2 tall, 2 short. So phenotypically, we have 2 tall, we have 2 short. Genotypically too, 2 tall, 2 short. So the phenotypic ratio is 2:2, because we can see physically 2 tall and 2 short. And also when you look at it genetically, considering the 2 letters, we still have 2:2. So the phenotypic and the genotypic ratio, when we get to F2 generation, is 2:2 when you backcross it. So backcross means you should cross an offspring with the homozygous recessive parent. And finally, on the terminologies, we have what we call Testcross. Testcross. Testcross is a cross that we use to determine the genotype of a particular organism that is unknown. The genotype of a particular organism that is unknown. For instance, we can see that a flower is red, meaning there is a dominant gene for red color in that plant. But we don't know, don't forget, each character is controlled by a pair of genes. We don't know if the gene of that flower is red like this or red like this. We don't know if it is homo red or hetero red. So when you are asked to find out that, that is when we use Testcross. And how do we do that? We cross it with a recessive color. Now this is red. We don't know the other letter. But we now cross it with, okay, other flower white. Let's say we have a white flower. We want to use it to get the genotype of this particular red flower. And we use these letters to represent white. You must use a recessive one. Take note of this. When you're having your genetic cross, except you are given the letters to use. If you are not given the letters to use, you must indicate before you start doing your cross the letter you are using for each cross. Or else you confuse the examiner and that may cost you your mark. And secondly, when you're having your genetic cross and you are not given the letters to use, be careful not to use some letters that are confusing. For example, if you are not given a letter, don't choose a letter like S, because we don't have a distinct difference between the uppercase and the lowercase. It is easier to work with letters like A. You know, the uppercase and lowercase, they are completely different. There is no way one will confuse that. Or B. Be careful not to use letter like C, because they are not too distinct, the uppercase and the lowercase. So R, for example, is also an interesting letter that you can use. D, all these letters there. Now, Testcross means we should do this. Now, we know that if a flower is red, the flower can either be homo red or hetero red. So you pick them one after the other, you cross them with the white that is recessive for you to get the actual genotype of this flower. Now, be that as it may, we now look at the last item on the objective, Mendel's work in genetics. Now, when we talk about Mendel's work, we are looking at the contribution of a particular man called Gregor Mendel. Gregor Mendel was an Australian monk. He worked so much on this concept of genetics, and he was given the name, the father of genetics. So he was performing an experiment. He tried to perform an experiment in order to know how traits are transferred from one generation to another. I take that again. Gregor Mendel performed experiments to find out how traits are passed, how characters are passed from generation to generation. And in order for him to get that, he conducted 2 different experiments. And he called the first one monohybrid inheritance. Monohybrid. As the name implies, mono. We know mono means one. Monohybrid. We have come across the word hybrid. A cross between 2 organisms of the same species but different genetic makeup. When we did something like this, TT, we said it will give us an hybrid, a mixture of that. Now, monohybrid means the crossing or the experiment on only a single feature, a single character. And what did he do? He tried to find out how traits are passed from generation to generation. So he did this. He got a garden pea plant. Garden pea was what he used, and he just picked one character from it, which is color, or let's say height. Just one. And when he did that, he picked that, he made sure that he picked pure breed, homozygous. For example, he collected tall plants of that garden pea and also short ones. Now, the question is, how did he know that they are pure breed of tall or short? Now, once he collected that, the seed of tall plants of garden pea, he separated it in a controlled environment whereby there would not be natural pollination. And he was planting that tall plant. Once they produce seed, he would replant it year in, year out for many years. When he discovered that for many years they were still producing tall plants, then he concluded that that means there is no gene for shortness in this particular one. So he concluded that this, he labeled that one tall. Then he collected the one for short and was planting it year in, year out for many years. And after planting it for many years, he discovered that all the offsprings, they were still tall. None was, not single one was tall. So he concluded that these are pure short plants. So he called that one short. Then he now performed the other thing by crossing them. Okay, now I have pure ones that are tall, and I have pure ones that are short. What did he do? He crossbred. He allowed them now to pollinate each other. Unlike the other one, when he separated them so that they would not mix up. Then, what was the result? He got this. TT. By now, you should know what the offspring will be. We have been doing this. Then we have this. Then we do for the other one. It saw that all the offsprings were still tall. Don't forget, this is F1. It did not stop there. It went ahead to do the second generation. And how did he get the second generation? He picked 2. You can see they are all the same. They are all heterozygous tall. So he just picked any 2 from them to get the F2 generation. Now, picking any 2 of the F1 offspring will give us this. Then he crossbred those ones too. Then we have TT, 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 TT. Now, no, yeah, this should be TT. He saw that in the F2 generation, he got 3 tall plants, 1 short plant. So he discovered that, okay, that means traits, characters are actually passed from parent to offspring. Although it may not show immediately all of the characters. The dominant ones may show first, but later on, the recessive ones will show forth in generations to come. And that's why you can see daddy is tall, mommy is tall, and you'll be wondering, why should they have a short child? Is this an illegitimate child? As a student of genetics, the first thing you need to do is to inquire. Check the background. Check the grandparents. Probably there is a gene for shortness in the parents that they have now passed across to that child. That's the first experiment he did. And the second experiment that he did, he called it dihybrid. Di, as the name implies. 2. In the first one, he used just one feature, one character. Now, in this one, he made use of 2 features because he wants to see now that, okay, now I know that this is how traits are passed from parent to their offspring. Now, I want to know, if we have 2 traits, how are they passed? Can one influence the other? Then he used what we call dihybrid inheritance. He picked 2 different seeds. Now, he picked 2. First, the first one is round. The seed shape is round. It's considering 2 characters now. And it's yellow. This one is round and yellow. While he picked another one that is wrinkled and green. Now, someone may ask, why should we now use Y to represent green? Why not G? When you are having a particular trait, now you know it is color. We are dealing with color here. So it is, you must use the same letter. The only thing you can use to differentiate them is uppercase and lowercase. So this capital Y is standing for yellow, while the small y is standing for green. So he picked this. Don't forget, not only color now. In the previous one, he was talking about height, mono, 1 character. But this one is talking about 2. He picked it, a seed with round shape, yellow color. And another one, wrinkle and green. So he tried to crossbreed those ones to see. Now, because this one, round and yellow, and we have wrinkle and green. This one, from all indication, will only give us this kind of children. A child that is round and yellow, or a child that is round and green, or a child that is wrinkle and yellow, or a child that is wrinkle and green. So these are the 4 types of children that this particular combination can give us. Now, when you cross these ones, what are the offsprings like? That was what he did. Now, to use our genetic diagram, the cross will be complex. It's either we use that or we use what we call Punnett square. What do I call it? Punnett square. Punnett square is simple. Let me illustrate it to you. For example, the other one we did TT and TT. If you are using Punnett square instead of what we did before, all you have to do is this. Punnett square. This same, you want to cross this and this. So you write them here again. You have T, T. So we have T, the same thing, T. So you cross them, TT, this TT. Don't forget, where you have capital and small letter, the capital must come first. So, and this will give us, so this is using Punnett square, while the other one is using diagram. Now, Gregor Mendel made use of Punnett square to find the offspring of this dihybrid. And he got something very interesting. So we have this, don't forget, this is round and yellow, round and green, wrinkle, yellow, wrinkle, green. You put the same thing here. We put the same thing here. Yes. We put the same thing here. So he made use of this Punnett square And all you need to do is just put them together. You have RY, RY. So put the R's together, put the Y's together. Then this one, put the R's together, capital Y. So that's how to complete your Punnett square. Now, the monohybrid inheritance and dihybrid inheritance led to the formulation of Mendelian laws. Monohybrid inheritance actually led to the formulation of the law of segregation of genes. That's the first law. That was brought about by monohybrid inheritance, saying that, otherwise known as law of segregation, say that an individual, they possess two alleles, and a parent will pass one, only one, to his or her offspring. While the second law is the law of independent assortment. And that law states that each character behaves as a separate unit and is inherited independently. That is, the fact that he picked round and yellow seed and wrinkle and green seed did not give him only round and yellow. He got some, they are round, but not yellow. After, in the offspring, we had some round and green offspring. Now, we are going to quickly have our questions and answer session. So just stick around as we answer the following questions. Question number 1. Explain briefly the following terms: gene, hybrid, trait. Now, I'm sure you are thinking, gene was the first term that I explained to you, and I told you is the basic unit of inheritance. It is that part of the cell that transfers traits from parents to their offspring. Is that taken? Then the second one, hybrid. When we talk about hybrid, is a cross, or you say the offspring that you have when you cross two parents of the same species but different genetic makeup. Offspring that you have when you cross two parents of the same species but they have different genetic makeup. Like you have one that is homozygous and the other one is heterozygous. Like we have the capital T, capital T, and the capital T, small t. Whatever you have here, they are hybrid, the children you have under this condition. And the last one there says traits. Another word for traits, characters, features. They are those attributes that are passed from parent to their offspring, that are inherited. And I gave you example, the height, the size, the shape, the color. They are all traits. Is that taking? Sweetness, when it comes to fruit, is another trait that can be passed. Number 2, two heterozygous yellow flowers are crossed. Take note, 2 flowers, heterozygous yellow. I hope you can remember the meaning of hetero, different. So if you say that, let this, Y is the gene for yellow flower. And we say they are hetero. They are heterozygous yellow, 2 of them. Hetero simply means it is capital and small. So 2 of them, they are hetero. And the question says, 2 heterozygous yellow flowers are crossed. Using a genetic diagram, state the phenotypic and the genotypic ratio of the F1 generation. You write this, parents. Then the next one you come, you put gamete. You separate them because now you want to cross. This is, then you cross it. You have capital Y, then you cross the other one, capital Y, small y. Take this here, capital Y, small y. Capital must always come before small. Now the question says, we should use this to give the genotypic and the phenotypic ratio. When you look at it, physically, we have tall, we have yellow, yellow, yellow. Once you have capital, you have to go with that. Capital Y is for the dominant gene. So we have phenotypically, we have phenotypic ratio. We have 3 to 1. Physically, 3 tall and 1 short. Genotypically, we have 1 pure breed tall, 2 heterozygous tall and 1 short. So genotypically we have 1:2:1. Don't forget 1:2:1. This is 1 homozygous tall. These 2 heterozygous tall, and this is homozygous short. So the next question, we have state 4 transmitted characters in plants. I'm sure we have mentioned that before. Color, height, size, sweetness. They are characters in plant that can be transmitted. Size of leaf, size of flower, they can be transmitted from parents to their offspring. Number 4, phenotype refers to the dash of an individual. Pheno, physical. A says genetic makeup, B, actual physical appearance, and C, recessive allele. So the correct answer there is B, actual physical appearance. Then we move to question number 5. The idea that different pairs of alleles are passed to offspring independently is Mendel's principle of dash. Don't forget Mendel's explanation of experiment using monohybrid and dihybrid inheritance. The first one, monohybrid, led to the first law, which is law of segregation of gene, and the second one, which is dihybrid inheritance, led to the second law, which is law of independent assortment. So the correct answer to this question is D, independent assortment. When we talk about round and yellow, wrinkle and green, we discovered that later on the round left the yellow to mix with the green. So each character behaved distinctly, independently. And finally, in garden pea seeds, smooth seed coat is dominant over rough seed coats. With the aid of a diagram, determine the result expected if homozygous rough pea is crossed with a smooth seed coat plant whose parents were rough coated. It's also very simple. Just take note of the words there. These are the words you need to take note of in this question. Dominant. Which one is dominant? You are told that smooth is dominant over rough. So you have to choose. If you are using S, I have told you to avoid this. So let's say you want to use R as gene for smoothness, for smooth. Okay? And you want to use this as gene for rough. And it says that these particular ones, it said one is dominant over the other one. So we look at it. I think I need to use this. So we say, if we say this is dominant, that's gene for smooth seed. Why this is for rough? Gene for rough seed. So take note. That's the first thing you must take note of. The dominant one, that's why we have used capital letter for it, the uppercase, and it said it's dominant over. Then the question continues. It said, with the aid of genetic diagram, we should determine the result expected if homo rough, take note, homozygous rough now, homo, meaning the same, is crossed with a smooth seed. This is for smooth. Now take note, a smooth seed coat which has, take note of that word, which has what? Plants whose parents were rough. Now if this one is smooth, but we are told that the parents, they had rough coats, what that means is that from all indications there will be gene for that rough coat in this seed. So this will be used for its hetero. So we are asked to find the offspring they will have. So all you need to do is you have this and you have this. So these are the parents. So you go to the gametes, you have this. Don't forget to put your sign. The question says we should find out. Then you pick the other one to this side. We have RR, then take it to this side. Now, what's the instruction there? We are to find It said determine the result expected. So this is the result that is expected. If you look at it, phenotypically now, we have, this is smooth, though hetero, and this is rough, homo. So phenotypically, we have 2:2, and genotypically too, we have 2:2. That is our last question for today. Thank you for attending today's session. For further reading and reference, please select genetics lesson contents that is available on learnathome.ng. Do join us next week, same time, for another exciting session.